taking away the repressor or adding an inducer takes away the glucose effect. So that now makes us think about this very different. Somehow, glucose is not about affecting something separate from the lactose impact. It is somehow affecting the lactose induction system directly. And that's what we need to figure out. So the paper we read which was a follow-up to this paper by Nada et al. In 1996, is a 1997 paper by Kamada et al. In the Proceedings of the National Academy of Science. And in this, they're going to now take this one more step. They're going to try to really test the textbook model. So here's the setup. They know that the lac Operon promoter is not a great promoter. They know that CRP with cyclic AMP helps stabilize RNA polymerase binding to the promoter. So they asked themselves, if we could remove the requirements for CRP and cyclic AMP, what would the textbook model predict for the glucose effect? Well, first of all, let's think about how could you remove the requirement for CRP and cyclic AMP. Kamada et al. Came up with a two-step, a double mutant way of removing that requirement. First, they made use of a mutation previously known as the L8 mutation, which wiped out the CRP binding site just upstream of RNA polymerase. So CRP can no longer bind there, and therefore no longer help RNA polymerase. But they joined that with a second mutation called the UV5 mutation, which is a mutation in the lac Operon promoter, such that now this promoter works well on its own, and it doesn't need help from CRP for RNA polymerase to bind stably and transcribe. So, in this case, CRP, 1, cannot help, and 2, it doesn't need to help. Now, according to the textbook model, what would this double mutant do to the glucose effect? Well, since the textbook model said the glucose effect was all about CRP helping, then if we make it such that CRP cannot help and it doesn't need to help, the textbook model would say this double mutant strain would not show doxic growth. All right? So Kamada et al. In 1997, the very first thing they did was to test that idea. So they set up the double mutant strain. They grew it in glucose plus lactose, just like Monod did. And here is the result. In squares is the measure of growth. We see it go up, and in about 3 hours or so on the x-axis, we see it level off, and then it goes up again. We see dioxic growth. Look at induction of beta-lactamase. In the first part of growth, up until about 3 hours, it basically is no induction, and then when the strain starts to take off again, which we can interpret as when it starts to eat lactose, we see beta-lactamase induction. That looks pretty much identical to the dioxic growth curve that Monod got, except here there's no way that CRP can be involved because it cannot bind upstream of the lac Operon promoter, and the promoter in fact works well on its own. So this directly proves that that component of the standard model is not where the glucose effect is. Now notice what I'm saying. I'm not saying that in wild type CRP isn't involved in getting expression of the lac Operon. It absolutely is. CRP plus cyclic AMP in wild type E. coli stabilizes RNA polymerase and helps it bind so you can get transcription. What I'm saying is that's not the glucose effect. The glucose effect is somewhere else. So to figure out what that somewhere else meant, Kamada et al. Took this L8UB5 double mutant and they added some things to it. So look at this graph, kind of study it. Here's L8UV5 on its own. All we're going to do in this next one is we're simply going to add IPDG, an inducer. Now, first of all, what's odd about that? What's odd is the very experiment is being done with glucose plus lactose. We already have a inducer there, and yet we saw the dioxic growth. We add IPDG, a second inducer, and now look at what happens with the growth curve. 1, the growth is constant. 2, Beta-glycosides is high from the very start. Why did adding IPTG make a difference if we already had lactose as an inducer? Well, what's different about those two inducers? Lactose requires lac permease to come into the cell in order to induce. IPTG, smaller, hydrophobic, can cross the membrane on its own and induce. And now we remove the glucose effect. How do we interpret this result? If lactose, for some reason, could not get into the cell when glucose is around, and we know this because when we add IPTG, we can induce even when glucose is around, that suggests the glucose effect is somehow inhibiting lactose transport. All right? Let me say that again. The glucose effect is somehow inhibiting lactose transport. All right, let's look at their next experiment. Again, L8 UV5 double mutant in glucose plus lactose media. And now they're going to add a third mutation, a mutation in a gene called CRR. CRR encodes one of the two subunits of the glucose transporter. All right, it removes the cytoplasmic component of the glucose transporter. Look at the graph again. Growth is constant. Beta-glycosides expression already starts pretty high. If we can't bring in glucose, we can't inhibit lactose. So now we're starting to think about the glucose effect as somehow the glucose transporter somehow affecting the lactose transporter. All right? Last part of this figure that starts to nail down this story. L8UB5 in glucose plus lactose, but now we have added a second copy of LAC-Y, but this second copy of LAC-Y is not part of the lac operon. It is behind a promoter that's on all the time. So bottom line, we're making lots of permease all the time. Look what happens. The glucose effect goes away. So somehow glucose transport was inhibiting lactose transport, but if I make more permease, I can take away the glucose effect. It's as if there's a one-to-one impact, if you will, or something like that, of the more, if I have a certain amount of glucose transporter, it can inhibit a certain amount of the lactose transporter. Make more lactose transporter, glucose transporter cannot affect it, or not as much. Okay? So, based on these experiments and a couple of others, Kamat et al. Proposed a different model of what the glucose effect means. Here's our cell again, but now included a membrane as this black line near the bottom of the page. We see the glucose transporter, two components, sitting in the membrane. We see the LAC-Y permease sitting in the membrane. All right? When the glucose transporter is active, when glucose is coming in, that's when the glucose transporter, in this model, is somehow inhibiting the lactose transporter. All right? When glucose gets eaten up and it's not active, now lactose can come in, pull the repressor off, we get transcription. Now, how could this actually work? Well, we know something about how the glucose transporter works. Let me back up here. When glucose is brought into the cell, it actually is immediately, during transport, changed from glucose to glucose 6-phosphate. And that's because the cytoplasmic component, this sort of maroon circle here, actually takes a phosphate from ATP and puts it onto glucose as it comes in. So, when glucose is coming in, when you're transporting in lots of glucose, that cytoplasmic subunit is putting phosphate groups on glucose as fast as it can. The moment it's got a phosphate, it's giving it away. Get another phosphate, give it away. That means its steady state is mostly to be not phosphorylated because it's giving away the phosphate as fast as it gets it. However, once the glucose is eaten, that subunit is no longer giving it away, so it's mostly phosphorylated, and that's when it appears not to be able to inhibit lactose transport. So that suggests how there could be a switch. The cell is monitoring glucose levels by whether or not that subunit of the glucose transporter is mostly in a phosphorylated state or mostly in a non-phosphorylated state. Okay. Now, one more piece of information that I didn't talk about in class is that mutagenesis experiments showed that there's a key part of the lactose transporter, the lac permease, that its sequence is critical to the glucose transporter inhibiting it. And it turns out the part that's critical is on the cytoplasmic side, which is where you'd predict it should be if it's going to interact with that cytoplasmic subunit of the glucose transporter. And interestingly, the sequence of those two loops, cytoplasmic loops, is very similar to loops in other membrane transporters that are known to be inhibited by high glucose levels. So in fact, what we see here with the glucose effect on lactose transport may be how glucose is used preferentially to many sugars. All right? And with that, we now have a much better understanding of the total story of the lac operon. You are in fact closer to nature's truth.